pathogens have become a major public health concern. In several studies, the use of molecular techniques for detecting and typing foodborne pathogens provides reliable epidemiological data for tracing the source of human infections. For detecting, speciating, typing, classifying, and characterizing foodborne pathogens of great importance to humans. A wide range of molecular techniques such as pulsed field gel electrophoresis, random amplified polymorphism deoxyribonucleic acid sequencing, repetitive extragenic palindromic, multiplex polymerase chain reaction, multi-locus sequence typing, and many others have been used to improve our understanding of key sources of foodborne pathogens, the source of infection disease, and species variability. Molecular techniques have the advantage of less laborious and more sensitive and efficient than traditional methods. Based on this article, we discuss about some commonly available molecular techniques, the advantages and usage to detect, characterize and type foodborne pathogens isolated from ducts and duct-related samples.
The most extensively used molecular approach for rapid detection, identification, and distinction of foodborne pathogen is undoubtedly polymerase chain reaction or PCR. The PCR procedure consists of 4 steps: collection, preparation, amplification, and post-PCR cleanup. During the amplification process, the PCR machine procedure takes place. It starts with a segment of a DNA sample which is inserted into an appropriate tube with a reagent and chemicals. The tube is inserted into the PCR or thermocycler. The thermocycler processes the solution into 3 steps: denaturation, annealing, and extension. Denaturation. A thermocycler is used to heat the solution contained in the tube to at least 94 degrees Celsius. The heat destroys the original DNA sample's hydrogen bonds and divides the DNA into single strands. Annealing. The sample mixture is then cooled to 50 to 60 degrees Celsius, allowing the DNA primers and the DNA polymerase enzyme to attach to the heat-separated individual strand of DNA. The nucleotides from the supplied mixed solution will now pair with the individual split strand of DNA produced by the heating process. Extension. They combine to generate a new complementary strand of DNA. As a result, from each of the single strand of the original sample molecule, a new duplicate double-stranded DNA molecule has been produced. The temperature fluctuates. The cycle is then repeated 35 to 40 times using the thermocycler, which automatically repeats the process of heating and cooling cycles. Each time the cycler performs a heating and cooling cycle, the resulting DNA sequence is doubled. After 35 doubling cycles, what began as a single short fragment of DNA from one sample can be amplified to generate millions of copies. And then, when the PCR process is finished, the amplified segment produced can be compared to other nucleotide sequences from a known source. In a separating gel, the PCR-generated nucleotide sequences are placed next to known nucleotide sequences from human, pathogen, or other sources. The gel is then passed through an electrical current, and the varied nucleotide sequences inside it generate bands that resemble a ladder, based on their electrical charge and molecule size. This is known as gel electrophoresis. Bands or ladder-like steps that migrate to the same level in the gel demonstrate the nucleotide sequence similarity. This is one of the most common methods for completing PCR tests.
Pulsed field gel electrophoresis, or PFGE, is a technique used for isolating larger pieces of DNA by applying an electrical current that changes 3 directions in a gel matrix. In PFGE, intact chromosomes are digested with restriction enzymes to produce a series of DNA fragments of varying size and patterns specific to a specific species or strain. Next, PFGE is considered a gold standard because of good reproducibility, typeability, and discriminatory power for foodborne pathogens. But PFGE is susceptible to genetic mutations, has limited availability, and takes longer time to complete a test. During the PFGE process, DNA degradation can occur, rendering those strains untenable. However, this can be resolved by modifying the PFGE standard procedures.
DNA sequencing is a technique involving the arrangement of bases of nucleotide include adenine, cytosine, guanine, and thymine. This technique is usually done after PCR technique with the purpose to increase the target gene. DNA sequencing is used for recognition, categorization, and also grouping in suitable taxonomy of pathogen. This technique is reproducible. However, it has limitation in terms of cost, accessibility, and duration of the result to be done. In the United States, food regulatory and public health authorities are increasingly using whole genome sequencing to aid in the discovery and foodborne pathogens incident regulation. Whole genome sequencing is a technique of sequencing which involves entire genome. It allows more rapid and effective outcome in response to foodborne illness.
One of the most popular methods for straight-level typing of microorganisms is multi-locus sequence typing (MLST), which is a technique in molecular biology for the typing of multiple loci using DNA sequences of internal fragments of multiple housekeeping genes to characterize isolates of microbial species. MLST approaches have been defined and adopted for many prokaryotic and eukaryotic microbes due to its simplicity and resolution. Plus, databases of thousands of MLST profiles and sequences are now available for a large number of microbial species, most of which are opportunistic pathogens. MLST method is an effective approach for the characterization of complex microbial communities as it skips the time-consuming isolation and cultivation steps. Other than that, it has high unambiguity and transferability of sequence data. It is also reproducible and scalable and provides good discriminatory power to differentiate isolates. On the other side, the limitation of MLST is that it is expensive and laborious. The selection of housekeeping loci requires reference genomes, and some pathogens are not suitable for this method.
Significant rapid techniques for the sensing of illness-causing microorganisms, which is molecular techniques, is precise and accurate. Nowadays, the molecular techniques usage in food industry and food safety microbiology is rising. Technological advancement is extremely beneficial to the national development sector. In order to achieve impressive results, the appropriate technique should be applied. It will enable the proper spending of costs, time allocation, accuracy, and feasibility.